Right, so Janine has alluded to these treatment systems that we're looking at. For the COHA Project 8, we're actually using the hybrid treatment systems and pilot treatment systems that we had from a previous project to look at the removal of PGRs and pesticides from recirculating water. So what I really want to talk about, I'll just briefly do a quick recap of what the objectives and schedule and what the hybrid treatment systems themselves are and how they're set up and how we did our bioassays. But I want to focus on the batch runs and the results because that was this year's work to get that going. So first of all, I love this survey that came out just actually after we had gotten approval for this project, and it was a survey that was done in the States by Sarah White and co. And what it says, this table says, is the contaminants of most concern by growers were actually pesticides and PGRs, and came up top of the list, which actually surprised me. I thought it might have been plant pathogens, but so pesticides, plant pathogens, and nutrients, that's sort of 1, 2, 3 and on the hit list. And so that's really what we've been looking at, all of those for the treatment systems. So the objectives for this project were to use the a couple of existing pilot systems that we had to assess the ability of the media to remove PGRs and pesticides, to look at the effective treatment sequences and to look at the effective operational parameters, and then to do KTT. Our KTT was a little short this year because of COVID, but like everybody else's, but we actually made quite a lot of progress in the whole thing. A loose definition of a hybrid treatment system. Really, we're considering it a site-specific treatment made up of a combination of media and hydraulic characteristics that are designed to treat water to meet individual farm requirements. So it can be site-specific. It's a modified constructed wetland design, and it's not a vegetated system. So this is what they look like. So our pilot systems, they're in 2 shipping containers. This is what it looks like inside. It's a series of a feed tank and 4 cells that we process the water through. And our original studies were to look at the removal of nutrients and plant pathogens, which they handle really quite well. And the current way that it's set up, this diagram shows there's the supply tank, and then there's a series of 4 treatment cells. The first treatment cell in each of them, one is called gold, one is called silver, because for whatever reason, that's what they were labeled. The tanks were labeled, so we carried on, or the containers were labeled, so we carried on with that. So the first cell is always the wood chips, and then followed by a series of other media, pea gravel and slag. We have that in 2 of the tanks. There's a pea gravel tank, wollastonite, a filter sand. And for this project, we flipped out one of the filter sand tanks and put in granular activated carbon, because we know that will remove the PGRs and pesticides. So that's a tried and true treatment. So that's kind of our gold standard in this whole system. So we've got that. We know we can use that to take everything else out, and we want to compare the other media to that. So the schedule for this year is highlighted in red here, 2020, 21. So this year the focus was on these batch studies. We carried on with some of the bioassay testing as well. And then we are also, there's 3 permanent systems out there from a previous projects, and we are carrying on monitoring those monthly to see how they're doing, because what we really want is long-term data. Currently, what we're monitoring those for pathogens and nutrient removal, but we might if we can squeeze it out of the budget, I'd like to actually do some PGR and pesticide work on those too, but we'll see how that goes. But this year was batch studies, lab analysis, and the bioassays of the effluents. So very briefly, we're doing this work down at Walden Gardens, and they've helped us tremendously. It's been wonderful. The bioassay work that we started doing it in the greenhouse, but we had to switch that this year, and sort of brought it home well, we just brought it home. Anyway, the method itself is broccoli germination and growth over 14 days. So you can see these are 7-day seedlings with the control on the left and the treatment on the right. This is a bonsai treatment, and so you can see the difference. This is what a whole bioassay looks like. So it's a lot of work. Again, this is bonsai from a control on the far left to about 0.2 milligrams per liter on the far right. So you can actually see the changes there. The assay itself measuring the hypoglottal length. And so after a few different attempts, this is what we came up with to do this most efficiently. And so Janine again alluded to our daughters helping us. And so we had Elizabeth and I we were our own little COVID unit this year. And so we got to work quite closely together, and we're still friends. So that's good. Anyway, so what we did was one of us I would cut the top and the roots off the little seedlings and pass it over, and she'd put it on a piece of sticky tape and line them all up and then measure and record those. And so in fact, we ended up with this visual representation of what the hypoglottals looked like. And so you can see the control on the left-hand side here and compare that. You can see the hypoglottals basically generally shortening up all the way across. And if you want to plot that, this is what it looks like. This was actually a method comparison, and we ended up using the method A, which is the darker bars here. That was our preferred method, and so that's what we carried on with. But you can see even here, even at, so this is about 3 ppb, so 0.003 milligrams per liter, so 3 ppb, we still have significant reduction in the hypoglottal length. And that's important when we look at the next slide. One of the other things that we did was to try and figure out what sort of levels things were at in the greenhouse. And so when we submit these samples to the labs, we have the University of Guelph lab. We have they have 2 scans and measure something like 400 different compounds. And so this is what we found out of all that. This is what we found in the off-bench runoff and in the return pit and the cistern. So this we worked in March. And on the right-hand side here, this is the feed water that we actually used in our batch tests. So what you can see, there's a whole range of things here, but the PGRs are highlighted in yellow. And you can see that there is frequently there's significant amounts of these compounds in that return water and even in the tank water, in the influent water. So this is the water basically that is recirculating. The yellow highlighted numbers on the right here for the August and October, those are numbers that we actually spiked. So for our batch studies, we spiked in the PGRs into the system. So those numbers are not floating around in the greenhouse. They're higher than normal, but if you look at fludioxonil, they're not way out of range. Our paclobutrazol or the bonsai was certainly higher than you would normally see. But if you look at the return pit in March, you see 3.6. These are all in parts per billion. So this is the level that you saw in the previous bioassay, that level was caused significant reduction in growth. So we're working within the range of what is around in the greenhouse. So these are our pilot plants. We moved them down to Walden probably a couple of years ago now. And they plumbed in a line from the recirculation tank. So this pipe here goes, pumps water into one of the systems, into the feed tank in the silver, and then we pump it over into gold. Here's what they look like inside again. And so that's where we ran our batch study. So fortunately, because of COVID, we weren't going into the greenhouse, but because these were outside the greenhouse, we were able to carry on and do what we needed to do. So just very briefly, the batch runs, so that the recirculation water went into the tanks, and we spiked them with a PGR mixture. We used Bioassay, Medallion, Cyclocell, and B9. Now Medallion and Bioassay come out in that pesticide scan. Cyclocell and B9 do not. But I haven't showed you, but we also did bioassays with these, and you can see that that with the Cyclocell and the B9 also inhibit growth, especially the B9. But they don't come out in that scan. So that's one of the reasons we have to do the bioassay, is to catch those PGRs that do not come out in the scan. And then the tanks were, all of the separate tanks, media treatment tanks, were filled with the spiked nutrient water, and we sampled after the final tank filling, considered that time 0, and then we sampled over the next 6 days. The samples were sent off for lab analysis, but in order to get our discounted price of, I think, $189 or something like that dollars per sample, we froze them and submitted them in big batches to the labs. And we also, at the end of 6 days, took samples for bioassay and nutrient analysis. And I won't go over the rest of the details. This is sampling. We sampled from the bottom of the tanks periodically. Before each sample was taken, we drained 10 to 20 liters out of the tanks in order to sort of move a fresh batch of water down into the sample. So there's Elizabeth. As Janine said, we were really lucky to have hired our daughters for technicians. And yeah, so there's the sampling. And so here's the results. So these are the lab results. The top 2 graphs, I hope you can read this. I hope nobody's looking at this on a cell phone, because that would be awkward. But the top 2 graphs are the summer run, and we did another run in the fall. So those are the bottom 2 graphs. This is the result for paclobutrazol or Bioassay, which is one of the most common PGRs out there, it seems. The dark bar on the left is the input. So that was the influent from the white tanks into the rest of the next 4 cells. And you can see very quickly, even in the afternoon of that, in the summer, in the afternoon, this is about 3 hours after filling, that we aren't seeing the paclobutrazol come out of the wood chip tanks. So that was really encouraging. Some of these early numbers here might be a little lower than they actually are, because part of this, you're driving, so we've rinsed and drained all the tanks before we start, but you're still driving a bit of water. There's always a bit of residual water in the bottom of the tanks, and you're driving that out. So some of the early numbers might be a bit low, but regardless, that it never showed up again out of the wood chip tanks. The P gravel was kind of low. We have a bit more variability than I would like in the influent going into these, and I think that's because of the mixing. So when we redid it in the fall, we did a lot more mixing to try and make sure that we had that spiked sample. And it's not an easy job to mix I forget how many 1,000-gallon tanks thoroughly. But I think we're better at that now. But regardless, you can see that there's a rapid drop in a lot of these treatments. So especially in the wollastonite and in the filter sand over in the silver one, but we don't see that same reduction in the pea gravel and slag tank. So most of our tanks are taking stuff out. Those two didn't do all that well when we talk about paclobutrazol. Now in the fall results look a lot the same. So the August ones were done, it was really quite hot. In the fall, I think we're running about 10 degrees here. So this demonstrates the effect of temperature. Again, our wood chips are doing a really good job. There's still a bit remaining in the pea gravel. The wollastonite isn't performing quite as well, and neither is the filter sand as it did in the summer. That might be a temperature thing. And again, the pea gravel slag tanks aren't taking out that much of the bonsai. And we have a lot of other results for the other chemistries that I won't show you all the graphs for those right at the moment. But the one thing I did want to show you, and this was interesting, whoops, there we've got some more highlights, right? In the summer run, our activated carbon, we did it in a layer on the filter sand. So we had about a 6-inch layer on the end, and I think it worked better than it shows that it did, because we had a layer on the top of the tank, we had to sample just underneath the layer, and I think that sampling was not as accurate. So we had to use a syringe and a tube, and so it wasn't a great methodology. So we changed that in the fall to a tank, and we had a small tank in the fall. The activated carbon takes out all of the paclobutrazol. The other graph that my internet's unstable. I'm going to turn my video off, I think. Can I do that? How do I do that? Stop video, just because our internet is not brilliant. And that might help it. So,
dragging a bit. We can hear your voice dragging a bit once in a while, but it's all good. We can understand what you're saying.
Okay, well, with the video off, it might help. I think it's our internet here. So the next slide here is the results for Phlonicamide. Now it's a pesticide, it's an insecticide, and the starting numbers are not very high, but what I wanted to point out was this really interesting thing for it. Most of these cells, the wood chip took some out, but the pea gravel and the wollastonite and the GIC layer, as I said, I don't like those results because I think that's a sampling issue. But also the filter sand didn't do a great job. So none of the other cells did a great job except the pea gravel and the slag. So in this case, those were the two treatments. That was the treatment that worked best. And it turns out that Phlonicamide is not stable at a high pH, and the pea gravel slag set, of course, has a very high pH. So it was broken down in those cells. And so I think that's a really interesting result and shows the benefit of having a treatment train. So if you're trying to remove everything, you need to focus not just on one treatment, but a treatment train works better. And that's the way we're running the hybrid treatment systems anyway. And so I think that's really good, because so we're seeing, depending on the media, you get different things taken out. Now, if you look in the fall, the activated carbon, of course, it also takes out all of the Phlonicamide, but an activated carbon system, like a regular system inside a greenhouse, is a very expensive proposition. And so if you can have the same treatment system taking out your nutrients or your pathogens and your PGRs and pesticides, I think that's a bonus. So I'd have to say I'm fairly happy with the results so far. Right, so
5 minutes, man.
Pardon me?
Got about 5 minutes.
Okay, so this is just a summary to look at all the chemistries. And the wood chips are doing a great job, except for example, this Phlonicamide. Filter sand and wollastonite did a good job in the summer. But the pea gravel and slag took out this odd chemistry here. In October, they look very similar. The filter sand and wollastonite did not do as good job. The pardon me. The GIC, or the activated carbon, took out everything as we would have expected. And whoops, sorry. Again, the pea gravel slag was good for the Phlonicamide. I'll just run through. Oh, this is doing bioassays during COVID. You take up your garage, you take up your office, you take up your daughter in your dining room, but you get your bioassays done. And I just wanted to point out this: the root systems in these bioassays were phenomenal, and they varied a bit with the PGR, but I just wanted to show you that picture because it's very impressive. Again, we see similar results here in the bioassay. So input, the wood chips do a good job. This is a bit lower than I would have expected, but the filter sand is good. Again, this wood chip, the one in the gold, was fine. So these are not significantly different from the control here, but the influent and pea gravel slag, so these are about 50% inhibition for most of those. Again, the October looks very much the same. So we have good control in wood chips and in the activated carbon. In the fall, again, these are filter sand and wollastonite are not quite as good as they were in the summer, and that might be temperature related. And the others are there's not a lot of difference. So just in summary, our reductions, again, influent is about 50% reduction, but the wood chip recovers that, 50% in these, wollastonite and filter sand. So it's a repeat really of what we saw in the chemistry. So we're seeing the same thing. And so this is covering those other two PGRs that we can't see in the lab scan. So the next steps are, this year we're going to look at the series. So we'll instead of looking at each one of those individually, we'll set up everything in tandem and look at the effect of the series runs, the media sequences, and the key operational parameters. And we'll do very much the same thing. And we have a few other plans for the permanent site, but we'll see how that works because I don't have time to talk about them. And thank you to everybody who's involved. There's a great number of people involved. We have a big technical advisory committee and support from Flowers Canada and Landscape Ontario, the OGVG, etc., and all our growers. So that's it. Is there any questions?